Hello everyone and welcome. In this video we'll be checking out February March 2023 9700 variant 2, of course, MCQ. So if you have any specific questions that you would like to check out in this variant, I have timestamps in my comments, and if you have any specific years that you would like me to solve, feel free to recommend those in the comments down below. Let's get started. Number 1. The photomicrograph shows cells from a human blood smear. Which calculation shows a correct method to calculate the actual diameter of the neutrophil shown in the photomicrograph in micrometers? Now, in order to calculate the actual diameter, you have to remember the triangle. It's important for calculating the diameter, the magnification, also the actual, and the image length. So, first off, it's I for image length, A for actual length or diameter, and M for magnification. So in this case, if we want to get the actual diameter, then A will equal to I over M. So in other words, it's the length of PQ divided by the magnification, which they mentioned was basically, they didn't write it down, but it's here in the answers. So of course, basically the length of PQ, but of course you're going to measure it in millimeters, never in centimeters. So if one of the options here said centimeters, make sure to cancel that out because they don't accept centimeters in Cambridge. And of course, basically, in order to convert from millimeter to micrometers, which is what they want, you multiply by 1000. And if you want to convert from micrometers back to millimeters, make sure to divide by 1000. Therefore, the answer should be the length of the line PQ in millimeters times 1000 divided by the magnification of the photomicrograph. Okay, so this is a tricky question, but it's also very intriguing. A student calibrated an eyepiece reticle using a stage micrometer. Each division of the stage micrometer was 0.01 millimeters. Okay, using with the times 10 magnification objective lens, 10 eyepiece reticle units match 10 divisions on the stage micrometer. Now, the same microscope was used with a times 40 instead of a times 10 magnification objective lens to measure the diameter of the alveolus. Interesting. So let's immediately match up the stuff that we have so far. One stage micrometer unit is 0.01 millimeters, and 10 stage micrometer units match up with 10 eyepiece reticle units. Therefore, we can assume that one eyepiece reticle unit is the same length as, one stage micrometer units under the same magnification. Alright, but now you change that to be from times 10 to times 40. So basically, if we were to imagine like what we would see something like this. Like it used to be 1, 2, 3, 4, 5, 6, 7, 8, and blah blah blah. Right, occupying like this same number, right? But now you change the magnification, so we literally zoomed in. Now we know that the eyepiece reticle is only placed onto the eyepiece lens, and it's not placed on the slide, and so it doesn't really get affected by changing the objective lenses. So that means basically that it will not be zoomed in. It's only the stage micrometer that's zoomed in. So after zooming in, that means basically that you're going to visualize much less of these divisions. And instead, let's say you visualize, an example, you visualize only one, right? So it's 4 times larger, because here from times 10 to times 40, that's 4 times larger. And so when this is 4 times larger, the length of the eyepiece reticle unit will obviously, like it used to be 1 to 1, now it's 1/4. Right? So now it's 4 times smaller. In other words, it's 1/4. In other words, you have to divide the length of one eyepiece reticle unit by 4 to get the length of the eyepiece reticle unit under this magnification. So it's 0.01 divided by 4, which is 0.0025, which is in millimeters. And of course, we're going to have to convert that to micrometers. So that's 2.5 micrometers. Okay, of course I multiplied by 1000 in order to get it in micrometers. So basically millimeters times 1000, Now 2.5, multiply that by 96, and you should get the length, or basically the diameter of the alveolus under this magnification, which is C. So I'm going to introduce another idea because some people are actually familiar with the way of using a formula, and maybe you'd find formulas interesting too because they actually help you out a lot when it comes to the eyepiece reticle. We know that there's this formula where basically we're going to be using the number of stage micrometer units, and we divide that by the number of eyepiece reticle units. And basically we multiply that by the length of one eyepiece stage micrometer. So length of stage micrometer units, and multiply that by 1000 because it's usually given in millimeters, and we want to convert that to micrometers. So that's just like a rapid formula. And if we rely on what's given to us here, then we can immediately say, okay, it's 0.01, and then basically we can immediately say it's 10 divided by 10 times 0.01 times 1000, which should give us 10, just like the previous one. Alright, but now we know that it's changed magnification from times 10 to times 40, and so the length of the eyepiece graticule will be 1/4 of that we assumed. So we're going to divide this immediately by 4 to get 2.5, and then 2.5 multiply that by the number 96, because basically you multiply that by the number of eyepiece graticule units occupied by the alveolus, and that is the other way. I'm going to give you an example just in case you're interested. So because sometimes he would give you, let's say you want to find the length of a nucleus, right? So in order to find the length of the nucleus, let's say the nucleus occupied let's say 7 eyepiece graticule units, okay? And one stage micrometer was in front of 50 eyepiece graticule units. So that means basically that it will be 1, which is divided by the number of eyepiece graticule units, let's say 50, and then multiply that by the, again, length of one stage micrometer unit, which let me give you an example, 0.1 millimeters, and then multiply that by 1000 to convert it to micrometers, and you should get 2 micrometers. And we said that the nucleus occupied 7 eyepiece graticule units. Well, 2 by 7 equals to 14 micrometers. That's the length of the nucleus. Usually it's smaller, but this is the answer for this example. Hope you understood. I'm going to move on to the next question now. Which statements are correct for chloroplasts and also for mitochondria? They contain ATS ribosomes. That is wrong. They can transcribe the circular DNA. True. They can transit mRNA. True. They are enclosed by the membranes. True. So 2, 3, and 4. Just C. Number 4. Which cell structure is not usually found in cells from the root? I would suppose it's... So cell wall is actually usually found, and chloroplast is not found because it doesn't really undergo photosynthesis. Okay, dialysis visiting tubing is an artificial partially removed membrane with pore size of approximately 2.5 nanometers, extremely tiny. Glucose molecules have a diameter of approximately 1.5 nanometers and can pass through the pores in the membrane. What can pass through the pores too? So bacteria, obviously not, because they have size between 0.5 to 5 micrometers, which is like way too huge, 5,000 nanometers, you know. Hemoglobin, absolutely not. It's a polymer. It cannot pass through 2.5 nanometer opening. Ribosomes are 20 nanometers, so even ribosomes cannot pass. Fructose is approximately the same size as glucose, maybe even smaller, so D. What is present in all viruses? Ribose, deoxyribose, adenine, thymine. Now we understand that ribose and deoxyribose, they form either part of DNA or mRNA nucleotides, and so basically sometimes viruses could be retroviruses, meaning they have RNA, sometimes they are adenoviruses, maybe they have DNA, so that is no and no. Thymine too, because it's only found in DNA. If they also said uracil is only found in the RNA nucleotides, so that's also wrong, so the answer is C. Okay, so for this question, 7. To estimate the concentration of glucose in an unknown solution, equal volumes of a range of known concentrations of glucose were each mixed with the same excess volume of Benedict solution. After mixing, the solutions were placed in a thermostically controlled water bath at 9 degrees Celsius for 3 minutes. Now the unknown solution was then treated in the same way, and the colors of the known and unknown solutions were compared. What's the independent variable in this procedure? Dependent variable. This is dependent, and so obviously it is concentration of glucose, right? So that is A, independent variable. We have to focus if he said independent or dependent, because dependent will be the final color of the solutions. Okay, temperature is control variable, volume of glucose solution, another controlled variable. Number 8, which statements are correct for amylose and also for amylopectin? So they're both carbohydrate molecules. That is true. They are formed by condensation reactions. True. They are linear molecules. No, amylopectin is branched, and they contain 1,4 alpha glycosidic bonds. Correct. So that's 1, 3, and 4, which is C. 1, 2, and 4. Excuse me. It is B. Okay. So let's move on now. Which statement is correct for comparison between saturated triglycerides molecules and unsaturated triglyceride molecules of approximately the same molecular mass? So saturated and unsaturated, the statement is correct for comparison. So unsaturated triglycerides have more double bonds and fewer hydrogen atoms than saturated. That is actually true. Okay, unsaturated triglycerides have fewer double bonds. That is wrong. Unsaturated triglycerides have more double bonds and more hydrogen atoms. No, if you have more double bonds, then you're gonna look something like this. So basically, this is a chain where the bonds, each carbon atom should be surrounded by, four bonds. Okay, so if I said there's two here, then there'll only be two, like one a bond with hydrogen and another bond with carbon. So literally I'll have one less for each hydrogen, like one less hydrogen for each carbon atom. Therefore, to always have less hydrogen atoms. Unsaturated triglycerides have a fewer double bonds. Again, wrong. So the answer is A. The diagram represents a molecule from a cell surface membrane. So what description of one of the labels is correct? So for one of the labels, we have what you call the fatty acids at the hydrophilic end of the molecule. So let's see. A is actually, yes, it is a fatty acid, but it's not at the hydrophilic end. It's at the hydrophobic end. Hydrophobic end of the triglyceride molecule. This is a phospholipid because we can see there's only two fatty acid chains and a phosphate group right here. And D, phosphate group at the hydrophilic end of the molecule, definitely. C, again, there is glycerol, but it's not at the hydrophobic end, it's at the hydrophilic end because again, this is hydrophilic. Okay. So which row correctly shows levels of protein structure that can be held together by each type of interaction? We have hydrogen bonds. Hydrogen bonds hold the secondary, tertiary, and quaternary structures sometimes, but never the primary structure. The primary structure, which is the sequence or the specific sequence of amino acids in a polypeptide chain, is basically only maintained by the peptide bonds. Because if you have an amino acid, and then if you ever see such a structure like a bead, this is one amino acid, another. What is this bond? It's a peptide bond. What is this structure? It's a primary protein structure. You don't see any hydrogen bonds. You don't see any covalent bonds besides from the peptide ones. You don't see any hydrophobic interactions or disulfide bonds or whatever. Now, so hydrogen bonds we can say secondary and tertiary. And then for hydrophobic interactions, tertiary structure. For covalent bonds, primary and tertiary. That is true because for primary, the covalent bond is peptide bond. For tertiary, the covalent bond is disulfide bonds. These are also covalent bonds. However, hydrogen bonds and ionic bonds, they're not covalent, of course. Which molecules contain at least 3 double bonds, saturated triglycerides, hemoglobin, or collagen? Now, at least 3. Here we only have 1 actually, because if you draw a saturated triglyceride, then remember it is going to contain like glycerol. There will be an O. This is the ester bond, and it will look something like this. The only double bond here, so this zigzag shape represents the fatty acid chain, and here because it's saturated, that means there are no double bonds within the fatty acid chain, so it's literally only carbon to carbon to carbon, and then the hydrogens all over the place, no double bonds at all. Okay, if I wanted to draw an unsaturated one, I would say something like this. Anyway, it is saturated, so the only double bond here is this one. So actually, yes, we have at least 3 dry triglycerides, 3 double bonds. So that is true. It contains at least so it is true. The triglycerides contain at least 3 double bonds. So we can do a check here. For collagen has lots of amino acids. An amino acid has a structure which is something like this: hydrogen, and then R group, hydrogen, and then this is the carboxyl group. So we can see there's one double bond in each one of them, because if you wanted to draw the peptide bond, it should be something like this. And we know that collagen is a polypeptide, so it has more than 3 amino acids, so obviously it has more than 3 the double bonds. So we can do a check here. Same thing for hemoglobin, therefore the answer is D. Okay, which diagram correctly shows hydrogen bonding between 2 water molecules? Obviously, the first one is wrong because of the signs. It's meant to be something like this. The delta sign should be present because this represents a strong positive and negative charges. However, the hydrogen bond is totally a weak bond, therefore it has delta positive, delta negative. And obviously we have to make sure of the distribution. Hydrogen atoms, they are never negative, they don't carry the negative charge, and that's because they only have 1 proton in their nucleus. If I were to draw a hydrogen atom, I would just for example hydrogen, and then for example if I want to draw the nucleus, it's only going to be 1 proton, 1 neutron, and 1 electron. That is going to be a hydrogen atom. right? However, if you draw an oxygen atom, it's going to have 16 protons. So if you place a water molecule which looks something like this, 1 oxygen, 2 hydrogen, right? Basically, if you place it somewhere in a tank, for example, where there are charges, obviously, the negative charges will all be attracted to the oxygen atom's nucleus. However, the positive charges will all be accumulating at the hydrogen atom's nucleus, because everything left, all the negative charges left and went here. Therefore, it's obviously going to be positive around here, and that is why it's going to be carrying the partially positive charge. However, the oxygen will be carrying the partially negative charge, and therefore the answer is B. For which statement describes an example of an extracellular enzyme, amylase extracellular enzyme is an enzyme that is produced inside the cell, but is going to be used outside the cell. So amylase in saliva is an enzyme that catalyzes the breakdown of starch in the mouth. That is true. Amylase is produced in your salivary glands, in the cells of your salivary glands, and then secreted into the mouth, the mouth cavity, and therefore it's an extracellular enzyme. So we can go ahead and check that. Let's look at the next enzymes. Carbonic anhydrase is an enzyme that helps in transport of carbon dioxide in the blood. The function is correct, but it's not an extracellular enzyme, because carbonic anhydrase is found is produced in red blood cells and it remains inside the red blood cells. DNA polymerase, again, it's not extracellular because it remains inside the nucleus, where it's going to be used during DNA replication. Same thing for RNA polymerase, it remains inside the cell in order to be used during transcription. Therefore the answer is A. Excuse me about that. A. Okay. Which row is correct for the enzymes that catalyze reactions using the lock and key hypothesis? Okay, remember lock and key hypothesis, it's just like its name. You have the enzyme which acts as the lock, and you have the substrate which acts as the key, and obviously basically they are complementary in shape. You can see this fits perfectly with this, and the enzyme is specific for the substrate, and obviously once they bind they form the enzyme substrate complex, and then this is maintained by the temporary hydrogen bonds between the R groups of the amino acids forming the enzyme and basically the substrate atoms. And of course, basically, if you compare this to the induced fit mechanism, this is the second mode of action that the enzyme could have. These are both theories, by the way. It's just suggestions of how the enzyme can work. So basically, it could be something like this. Maybe the enzyme has some weird and not fixed, like flexible shape at the beginning, and then it meets the substrate. Once it sees the substrate, it remembers its origins and it decides to change back to its original shape. And so sometimes also the substrate changes a little bit to allow the perfect fit between the enzyme and the substrate, and so this happens. And obviously this happens when the substrate enters the enzyme active site, and then basically there will be formation of the enzyme substrate complex, and then same thing, the temporary enzyme product complex, and then the product will be released, the enzyme returns back to its original shape, and it's ready to be reused. So let's look at the effect of the enzyme on the activation energy of the reaction being catalyzed. However, the mechanism of the enzyme, it is always going to lower the activation energy. Okay, the shape of the active site in comparison to the substrate, as we mentioned, it's complementary. Okay, before, during, and after the formation of the enzyme substrate complex, that is correct. Let's move on. So a scientist investigated the rate of breakdown of hydrogen peroxide. 4 experiments were carried out using different mixtures. The results are sketched in the diagram. So obviously substrate only, it's not going to be breaking down unless basically like temperature is high or something like that. And in the case of hydrogen peroxide, I mean, I can see why it's going to be breaking down. Substrate and enzyme, obviously it's going to have the highest rate of reaction. If there's an inhibitor, that's going to lower down the rate of reaction. But for competitive inhibitor, we have to understand its effects versus the non-competitive inhibitor. Now we know what the Vmax is. The Vmax is the maximum rate of the reaction, right? Now the competitive inhibitor is not going to lower the Vmax. You're going to eventually go back to the Vmax if you increase the substrate concentration, because we understand that the competitive inhibitor, from its name, it's competing with the substrate. If you increase the amount of the substrate, obviously the substrate is going to win, right? And so basically the maximum rate of reaction does not change. However, the affinity of the enzyme to the substrate is going to be decreased. What is the affinity, you say? It's basically the ability of the enzyme to bind to the substrate as soon as it sees it. Of course, when you see there's a distraction, like the competitive inhibitor, the enzyme is going to be distracted and it's not going to immediately bind to the substrate, unlike when the enzyme is alone with the substrate, right? And so we say the Km is what allows us to understand the affinity of the enzyme to the substrate. The Km is the substrate concentration at which half the Vmax is reached. So this is the Vmax. Here we have half the Vmax. Okay, basically, if we know that half the Vmax is this, then of course we can draw some type of line over here, and we can see the Vmax for this enzyme is lower compared to this enzyme. And we understand that for one does not have any inhibitor involved. It doesn't have any distractions. So it has a higher affinity for the substrate. So obviously, and it has a lower Km. So that shows that the lower the Km, the higher the affinity for the enzyme for its substrate. However, the higher the Km, the lower the affinity of the enzyme for its substrate. Let me write this down. Again, the higher the Km, the lower the affinity. The lower the Km, the higher the affinity, which you can see in the graph. However, the effect of a non-competitive inhibitor is just like the shape of graph number 3. Okay, so for graph number 3, you can see actually that the Vmax is never reached. Okay, so here, let's look at the Vmax and get half the Vmax, because as I mentioned again, at the substrate concentration at half the Vmax, this is the Km value. And based on our results, we were able to assume that as you increase the Km value, the affinity for the enzyme for its substrate decreases. So let's see the effect of the non-competitive inhibitor, which we now know the graph looks something like this. The Vmax is lowered on the Km value. So this is the Vmax for the enzyme with non-competitive inhibitor. Now we want to see the effect of the non-competitive inhibitor on the Km, because that's where it differs. The non-competitive inhibitor has no effect on the Vmax, however it has an effect on the Km value. Let's see if the non-competitive inhibitor has an effect on the Km. The Km again is a substrate concentration at half the Vmax, or half the maximum effectivity of the enzyme. And so if we know this is the Vmax, we can assume that maybe this is half the Vmax, you could say. Okay, however, if we know this is the Vmax for this one, let's say this is half the Vmax, so this is half, and I suppose basically then this is half the Vmax. I'm just trying to approximate it, it's not exactly true, but if this is half the Vmax, a line from here and a line from here, we can see that the substrate concentration for both is the same. Okay, so that means that the Km value for both is equal. So that means it has no effect on the Km value. So let's just summarize this. If you see a graph looking like this, the most normal, this is the enzyme alone, and a graph where if you increase the substrate concentration, you'll eventually reach back to the Vmax. This is the competitive inhibitor. And if you see a graph where it doesn't really reach the Vmax, it reaches a plateau, but the Km value remains the same, this is the effect of a non-competitive inhibitor. And so let's see what they want for this question, which row shows the correct lines for the two experimental mixtures. Substrate only, it's obviously 4 because it doesn't look like there's actually any much breakdown going on. So I would say it's 4, but substrate and enzyme and competitive inhibitor, we've established that it's 2. So that is B. Okay, so which of these substances can pass directly through the cell surface membranes without using a carrier protein or a channel protein? So things that can pass directly without channel proteins or carrier proteins have to either be lipid soluble or like an organic solvent, something that is hydrophobic or non-polar, or something that's extremely small, that even if it's polar, that doesn't really matter, like in the case of water, oxygen, carbon dioxide, and urea. So let's see what we have here. Mineral ions, obviously not, because they require carrier and channel proteins since they are hydrophilic and they are large in size. Same thing for glucose or fructose, whatever this molecule is. However, carbon dioxide is, as I mentioned, one of the molecules. Okay, what happens to the surface area to volume ratio of a cube when the length of each side is doubled? Actually, the ratio halves. Now, you don't need to know this. You don't need to memorize this. You can actually just experiment. You understand that the surface area to volume ratio is literally what the name, okay? You have to get the surface area in order to calculate it for a cube. It's just going to be 6s squared, and then basically divide that by the volume, which is literally side by side, or in other words, it's side cubed, okay? So we can, for example, let me give you the example of the cube with the surface area, or let's say the length of 1, width of 1, and height of 1, okay? So basically, of course, it's a cube, so it's literally just 1 squared and to 1 to the power of 3. That is 6 divided by 1, which is 6, a really high surface area to volume ratio. Now, let's say I doubled the side length, so let's say it's now 2. So it's 6 multiplied by 2, all of it squared, and then divide that by 2 to the power of 3, right? 3. So that halves. In the exam, if you do not know this piece of information, you can just calculate it. Okay, we're moving on now. We have which events are part of the mitotic cell cycle. Okay, again, mitotic cell cycle, it's actually, it includes everything, but when it comes to what's it called, the cell mitosis itself, actually it only includes prophase, metaphase, anaphase, and telophase. So remember the bar or the pie charts. So here we have what's it called, okay, I hope this, no, something, okay, something like this. So this is G1, this is a cell cycle, okay, G1, and then we've got S phase, right, and then here we have G2, second growth period, and then here we have mitosis. Okay, so mitosis is part of the mitotic cell cycle, okay, but the cell cycle is not part of mitosis. So if he was to ever ask you what is part of mitosis, you would only say PMAT, which is the phases that make up mitosis. There are 4 phases called prophase, metaphase, anaphase, and telophase. Okay, this is also cytokinesis. It's part of the cell cycle, but not part of mitosis. Therefore, all of them are parts of it. It's A. Now, the estimated total number of red blood cells in a human body is 2.5 by 10 to the power of 13. And it is estimated that each day 2.5 by 10 to the power of 11 red blood cells are removed from the circulation and are replaced by stem cells in the bone marrow. What is the percentage of total number of red blood cells is replaced each day? So what percentage is replaced each day? We just divide 2.5 by 10 to the power of 11 by 2.5 by 10 to the power of 13. Multiply that by 100 to get the percentage. So that equals to 1% of the red blood cells are replaced each day. Now, DNA polymerase catalyzes the condensation reaction between molecules during semi-conservative DNA replication. What is joined by DNA polymerase? This is a repeated question actually. It's nucleotide and nucleotide, okay? So that is C. And basically 2 polynucleotide strands make up DNA molecule. Which description is correct? The percentage of cytosine is 50%, that of guanine in the whole molecule. Actually no, the percentage of cytosine to guanine, the ratio of cytosine to guanine is 1 to 1. The percentage of cytosine is the same as that of guanine in the whole molecule. That is correct. Again, it's 1 to 1. The percentage of cytosine is the same as that of guanine in each strand. No, not in each strand, because they are complementary and hence they're going to line up against each other according to complementary base pairing rule. But they're not going to be the same on each strand. It's going to be different. Okay, the percentage of cytosine in each is the same in each strand of the molecule. That is wrong, because obviously the reason for variation between us is basically the fact that we have different sequences and different numbers of each of the bases, right? And so basically you cannot say that the percentage of cytosine is always going to be the same, okay? So the answer is B. Okay, now for this question, the PTX1 protein is found in a wide range of animal species where it is regarded as essential for normal metabolism. In sand rats, the gene coding for PTX1 protein has a much lower proportion of A and T nucleotides. A lower proportion, interesting. Okay, and basically than PTX1 gene from other animals. Now, finding the complete DNA sequence of PTX1 gene in sand rats has been difficult. Sequencing involves splitting off nucleotides one at a time from a single strand DNA. What could account for the difficulty in finding the DNA sequence of PTX1 gene in sand rats? So, I suppose the PTX1 gene is present in only a small proportion of nuclei. Actually, it's present in all nuclei, so that's wrong. PTX1 gene is transcribed in only some cells. That is true, but this does not make it difficult in finding basically the sequence. It actually has nothing to do with finding the sequence. It's totally fine to find some cells, so I think this is also wrong. And the strength of the hydrogen bonding between the 2 strands of PTX1 gene is unusually high. Now, this makes sense considering he said he has a lower proportion of adenine and thymine. And between adenine and thymine, we have only 2 hydrogen bonds. If you substitute those by cytosine and guanine, which have 3 hydrogen bonds, then obviously you're increasing the proportion of the number, you're increasing the number of hydrogen bonds, which makes it more difficult to split the strands and form single strand of DNA. I think that's the reason why it's so difficult. So, 23, that is D. Different tissues in a plant were supplied with radioactively labeled substances to identify which tissues were actively synthesizing mRNA. So obviously, which radioactively labeled substance would be suitable for this experiment? Anything that is specific to RNA and not DNA, because there are differences between mRNA nucleotides and DNA nucleotides, right? So DNA nucleotides have both of them are going to have pentose sugars, but DNA has a deoxyribose, while RNA is going to have ribose. That's hence the name. It's called deoxyribonucleic acid, right? So another difference could also be the fact that bases, right? So there's adenine, thymine, cytosine, and guanine in DNA, but however in RNA there's only adenine, cytosine, uracil, and guanine. There's no thymine. So what can we do in order to basically identify tissues that we're synthesizing with mRNA? We can have basically radioactively labeled uracil, right? And we can also have radioactively labeled ribose. Inorganic phosphate and adenine are present in both DNA and RNA, so that would be stupid because all cells have DNA. Okay, 2 and 4 only. C. Now 25. Which row correctly matches the structure and function of the phloem sieve tube elements? The structure, peripheral cytoplasm. Okay, the phloem sieve tube elements, let's go ahead and draw them. Okay, so basically, let's say this is the wall it shares with the companion cell. Okay, so basically here we have nucleus and mitochondria and lots of what's called cell structures. However, here we only have peripheral cytoplasm, not many cell structures. You can see maybe some smooth or rough endoplasmic reticulum, but no nucleus. So basically peripheral cytoplasm with no nucleus, correct function to provide as little resistance as possible to the flow. That is also right. End walls are modified to form sieve plates. It is a down rate of transport of solutes. Actually, no, in order to reduce the resistance and to allow the continuous flow of the solutes. And elongated cells join end to end, sure thing, why not, and to form a tube to transport dissolved mineral ions in water. The function here is wrong. This is the function of the xylem vessel element. Cellulose cell wall with no lignin, true. To prevent loss of water, absolutely not. Actually, lignin is what prevents the leakage of water. If you do not have lignin, you're not going to prevent the loss of water. And water is not the main thing that's being transported here. The important part is avoiding loss of the phloem sap. It's really precious. And hence, I'm going to be choosing A. Now, which feature of xylem vessel elements help adhesion during transpiration? Okay, so lignin forms a complete secondary wall. Okay, this does not help adhesion because adhesion is supposed to be between the water and the wall, right? So basically it's the bonding between the water and the side walls of the xylem vessel element or any tube. So lignin forming a secondary wall is not the right thing because lignin is impermeable to water. Okay, so lignin does not like water. Okay. New vessels carry extra water as the plant grows. There are no cross walls between the xylem vessel elements. I think this more helps with cohesion because if there's no cross walls, then basically water will not suddenly, stop flowing as one continuous column. Elements form a narrow tube. If they form a narrow tube, then it's going to increase the number of water molecules that are exposed to the tube walls. And that means basically that it's going to help with adhesion. I think it's D. Okay, mass flow is the bulk movement of materials from one place to another. How many vessels listed carry fluids by mass flow? Artery, phloem, sieve tube, and vein, and xylem vessel. All of them, of course, it's D. Okay, moving on. Cellulose, lignin, and suberin are components of various plant cell walls. Descriptions of these cell wall components are listed. A component of the Casparian strip is going to be suberin, redirects water into the symplast pathway. Another suberin, another one about suberin. And hydrophobic components of the cell walls, you can say lignin or suberin. And basically water interacts with this molecule in the symplast pathway, definitely cellulose. So which row correctly matches the three descriptions with the cell wall components? Cellulose, as I mentioned, is 4. Lignin, it's a hydrophobic component, could be 3. It could be, it's only 3. And then 1 could be, suberin could be 1 or 2. Now the arrow shows the direction of water movement across the plant's root from water in the soil to the stem. Okay, which arrow shows water movement only in the Laplace pathway? Okay, so Laplace pathway consists of two things. It's the intercellular spaces and the cellulose cell wall. So literally from its name, it's called the dead pathway. Okay, anything dead, anything not alive is going to be forming the Laplace pathway. Symplast pathway is going to be the alive pathway. Cytoplasm, cell surface membrane, plasmodesmata, like here, these are basically cytoplasmic extensions. Symplast pathway. There's also the vacuolar pathway, but lately they've been considering it more as symplast pathway. So the Laplace pathway is, now this is wrong because it passed through cell surface membrane and cytoplasm. This is also not it, no. And I think it's going to be 1 and 2 because we only see them passing through. But no, 1 doesn't, 2 is wrong. I think 4 is right, 2 and 4, which is C. Now moving on. Okay, number 30. Which row correctly identifies sources and sinks of sugars? Root cells absorbing mineral ions. I don't think it can be considered a source or sink. This is most. So Mineral ions are transported by the xylem vessel element. There is no source and sink in xylem, like the transport only occurs in one direction. Difference though in the phloem is that there's a source and sink, and that can change at different times of the year when the plant is growing, right? So the sink is basically what receives the sugars, and the source is what provides them. When root cells are absorbing mineral ions, it doesn't mean that they are supplying sugars, are they? That's the only time when something under the soil is going to be the source is when there's an endosperm and when there's a starch storage, right? Like in the case of the storage cells of seed that are starting to grow. These are sources. So basically, they are absorbing mineral ions, and that's by active uptake, right? And active uptake requires energy. And how do they get this energy? It's by respiring glucose. Where are they supplied with glucose? Well, that is where they are the sink. Okay? So that's why I'm going to choose A. Moving on. We have 31. A photomicrograph shows a section of throat tissue and with an artery. So which row correctly shows the type of artery and whether the blood inside the artery is oxygenated or deoxygenated? So an artery, and this is most so clearly lung tissue because of the alveoli that we can see over here. So obviously the artery is going to be pulmonary artery, and what we know about pulmonary artery is that it carries the oxygenated blood. It is a muscular artery, and let's see basically which row correctly shows the type of artery and whether the blood inside is so type of artery, as I mentioned, muscular and deoxygenated. So that's basically A. Heart surgery may cause a disease and a transmission of impulse in the Purkinje tissue to the right side of the heart. So what is a possible effect of this disease? So the impulse will be delayed by the atrioventricular node, I think, because like the decrease in transmission to the right side, that will result basically in like a weaker contraction by the muscles in the right ventricle. So let's see, the muscle of the right ventricle would contract slightly more slowly than the muscle of the left ventricle. Possibly, yes, the muscle of the right atrium would not contract as fully as the muscle of the left atrium. Really, the Purkinje tissue, if you know the structure of the heart, it's within the septum, right? So it has no effect on the atria. I'm sorry for the terrible drawing, but I hope you get the point that I'm trying to give. This is the right atrium, right ventricle. Here we have the sinoatrial node, the atrioventricular node, and then basically non-conducting band of fibers. So here we have an insulation, and the only way for the ways of excitation to pass from the atrium to the atrioventricular, to the basically ventricles, is through the atrioventricular node, then through the Purkinje tissue, which is right over here, to the apex of the heart, and then like all the way up to the walls of the ventricle, right? So it has no effect on the right atrium. It has no effect on like what's it called. The impulse will be delayed by that. Like this is already something that happens. Okay, this is like something natural. It's not part of the disease. The sinoatrial node will be transmitting fewer impulses. It has nothing to do with that. Okay, the transmission here, it's the problem with the Purkinje tissue. So obviously it's C. Okay, excuse me, I by mistake chose C. It's actually B. Now, the diagram shows a network of blood vessels that supply blood to the muscles in human body. So what is correct comparison between the blood at X and the blood at Y when the muscle tissue is at rest? So when the muscle tissue is at rest, blood pressure compared at X with the blood pressure at Y. So obviously the blood pressure here in the artery region is higher than the blood pressure at Y because it's like capillary and it's even closer to the vein. The water potential of blood at X compared to the water potential of blood at Y. So also water comes in with a higher water potential because here water will already be going out and into the tissue fluid. So I'd assume that it also has higher and higher. I think that's C. Okay, now for 34, scientists have shown that basically the oxygen saturation curves for hemoglobin are smaller mammals are to the right of those of larger mammals. What does this suggest about hemoglobin of smaller mammals? So hemoglobin of smaller mammals is going to have a lower affinity to oxygen, or like in other words, it's able to release oxygen more easily. Exactly. So this is letter C, but let's read the other options. At low partial pressures of oxygen, it binds to oxygen more strongly than hemoglobin of large mammals. That's wrong. To the right means it's shifted like this. So this is the graph, and then this is the smaller mammal. So it binds to less oxygen even at like the lower partial pressures, which is not suggested by this answer. It saturates with oxygen at lower partial pressures of oxygen than hemoglobin of a larger mammal. Again, no. It saturates the larger partial pressure of oxygen, and as I mentioned, C is the answer. But when the partial pressure of oxygen is high, it carries more oxygen than hemoglobin of larger mammals. No, they carry the same amount if it's high enough, but usually also it will carry more. The larger mammal will carry more than a smaller mammal. Okay, 35. What is the effect of an increased concentration of carbon dioxide in the blood? So carbon dioxide is going to decrease the pH and decrease the affinity of hemoglobin to oxygen. So increase the movement of chloride ions out of the red blood cells, grow the red blood cells. So here we have CO2, and then we have water, a reversible reaction, and then carbonic acid is formed. This enzyme here is going to be carbonic anhydrase. This dissociates to hydrogen ions and hydrogen carbonate ions. Hydrogen ions are going to be bonding with hemoglobinic acid. Going to bind with hemoglobin, oxyhemoglobin, to form hemoglobinic acid. The important parts for this question here: hydrogen carbonate ions are going to move out of this cell by facilitated diffusion through channel proteins, and in order to maintain a balanced polarization state of the plasma membrane, we have chloride ions coming into the cells. Therefore, this is wrong. Okay, it's supposed to move into, not out of. Increased concentration of hemoglobinic acid, that is true, because as I mentioned here, hydrogen ions will bind with oxyhemoglobin to form hemoglobinic acid, exactly. And decreased concentration of hydrogen carbonate ions in the blood plasma, no, as I mentioned, it's going to increase. Decreased concentration of carbaminohemoglobin, actually no, it will also increase, because another way carbon dioxide is going to be transported is by binding to the terminal amine group of hemoglobin in order to form carbaminohemoglobin. The answer is B. Okay. 2 of the requirements of an efficient gas exchange system are a large surface area and a short diffusion distance. Which row correctly describes how alveoli are adapted to meet these requirements? Large surface area. Okay, so in order to increase the large surface area, it has, of course, the alveoli, they're folded. That's how we know them. So yes, they are folded and interconnected. That is true. Short diffusion distance. We can say they're 1 cell thick, or let's see, an extracellular layer inside the alveolus contains what capillaries. This does not decrease diffusion distance. Alveolar walls are next to capillaries. Okay, I can see how. Let's look at this answer. Gas dissolved in a layer. This does not increase surface area. Okay. Walls of the alveoli are 1 cell thick. Totally. Red blood cells are very close to the capillary walls. However, walls of the alveoli are formed of squamous epithelial cells. Doesn't increase the surface area. C. What defines an infectious disease? Infectious disease, basically, they're deviations from health, and they're caused by a pathogen that could be transmitted from a person to another. So let's see. Symptoms are caused by bacteria, not necessarily. Let's see the other answers. Symptoms are caused by a pathogen that is transmitted from one host. Totally. Symptoms are caused by a microorganism that is carried by a vector, not necessarily. Symptoms are caused by a virus that mutates to infect a new species. Again, not necessarily. It could be caused by a virus, by protoctist, by yeah, so anything really, just basically any type of infectious disease, any type of pathogen that comes into your body, as long as it can be transmitted from one person to another and it's caused by a pathogen, that is an infectious disease. A non-infectious disease example is cancer, sickle cell anemia. A hurricane destroys a large town on an island. People move away from the town, set up tents, or sanitation is poor. Which disease is most likely to spread within a week of the change of living conditions? Because sanitation is poor, we're going to assume cholera. Cholera depends heavily on sanitation. Just like basically people who live in areas where there's low sanitation, people have terrible hygienic habits, like someone doesn't wash his hands before preparing his food, right, he's gonna end up transmitting cholera, right? That's basically the main thing. For TB, I would say, if they moved into tents and it was overcrowded, obviously TB. If they basically moved into a really humid place, I would say malaria. HIV, I'd say if they're not educated enough. That's 39. Which statements correctly explain why viruses are unaffected by penicillin? So here's the mode of action of penicillin for you to understand. There's this enzyme known as autolysins. Autolysins is an enzyme that breaks down the cell wall into basically blocks, and what's that's basically in newly produced or and young bacterial cells. Okay, so just for growth to happen. Okay, so basically it breaks it down into these subunits, and then there's another enzyme. We do not really call it names because they really change the names a lot, so we just refer to it as the enzyme that catalyzes the formation of hydrogen bonds between peptidoglycan subunits, right? Now, what happens, penicillin comes in, it's going to act as an inhibitor to this enzyme, and hence reducing or stopping the rate of the formation of the hydrogen bonds between subunits of peptidoglycan cell walls. Alright, so basically the viruses do not have cell walls, do not have cell walls made of peptidoglycan, and do not have the enzyme that catalyzes the formation of the hydrogen bonds between the peptidoglycan subunits. Hopefully that made sense to you. So the answer here is clearly D. Penicillin only blocks peptidoglycan synthesis. So which process characterizes the mode of action of phagocytes? Phagocytes, we have antibody production. No, that's the role of B lymphocytes, and specifically plasma cells that are produced by the differentiation of B lymphocytes. Receptor binding, okay, sure, because we have adherence. That's one of the things that happen in phagocytosis. Okay, we have endocytosis, true. Exocytosis, true. Hydrolysis, true also. The action of lysosome, 2, 3, 4, and 5, C. This was the end of this variant. I hope this was useful, and good luck everyone on your tests. Goodbye.